All right, today's lesson, we are specifically looking at anaerobic cellular respiration. So recall anaerobic means without oxygen. So in an environment where there is no oxygen. So previously, everything we've looked at, all the cellular respiration we were talking about after glycolysis, we looked at the Krebs cycle, the electron transport chain, and so on. That was all with oxygen. Okay, so that was called aerobic cellular respiration. All right, so today we are looking at anaerobic cellular respiration, which will also be called fermentation. So when oxygen is not available, some organisms are able to use those conditions of no oxygen to still produce ATP. They use a different final electron acceptor than oxygen. All right, so recall in aerobic cellular respiration, it was oxygen that was the final electron acceptor. All right, so in anaerobic, we'll talk about what the final electron acceptor will be. So the products are going to differ from those of aerobic cellular respiration. Molecules like lactate, ethanol, and acetic acid are the commonly produced products. As we go through this unit, this last bit of this unit, you'll see that this fermentation and anaerobic cellular respiration is very important in different industries. So for example, the food industry uses anaerobic respiration in breads and cheeses. The wine industry depends on it to create wines and beer and other alcoholic drinks. All right, so just a quick summary here of what we've looked at so far. Here at the top is photosynthesis. So we had the light reactions, and then we had the light independent reactions products were ATP and NADPH. And then we've looked at cellular respiration so far, where we had glycolysis was the first step, which produced a net of 2 ATPs as well as 2 pyruvates. And if it was continuing aerobically with oxygen, we would have had the production of acetyl-CoA into the Krebs cycle and then the electron transport system. We are looking at now glycolysis produces the ATP and the pyruvate, but there's no oxygen present, so we are going into this fermentation stage. All right, so like I said, anaerobic cellular respiration, it uses a different final electron acceptor than oxygen. All right, in aerobic, it was oxygen, so anoxic environments, so they contain no oxygen. So it usually includes an electron transport system and a concentration gradient to generate molecules of ATP, but this method is not as efficient as aerobic respiration. So it's going to provide less energy, meaning fewer ATP will be generated. So since oxygen is not available as the final electron accepting molecule, a different molecule is used. Usually it's going to be an inorganic chemical like sulfate or nitrate or carbon dioxide. All right, so those are the 3 common chemicals that are used instead of oxygen as the final electron acceptor. Common byproducts are elemental sulfur, nitrate, elemental nitrogen, and methane. So fermentation will occur typically in the cytoplasm of the cell, and it uses metabolic pathways that do not require oxygen or an electron transport system to produce ATP. Instead, there's going to be this process called fermentation. It's a metabolic pathway that includes glycolysis and 1 or 2 reactions in which NADH will be oxidized to NAD+ by reducing the pyruvate to other compounds. Like I said before, it is much less efficient at supplying energy than aerobic cellular respiration. It's only going to produce the same amount of ATP that was generated in glycolysis. So the two types of fermentation we're going to talk about. The first is lactate, the second is ethanol. So let's talk about lactate fermentation first. This type of fermentation occurs in some single-celled organisms and in some animal cells that are temporarily without oxygen. So like if you're working out really hard and you're kind of like running out of oxygen, you'll be using this lactate fermentation. So NADH is used to convert pyruvate to a molecule called lactate. This molecule is also called lactic acid, and it happens in a single step. So we can see that it happened right here. That pyruvate got converted to lactate in a single step here. And what you'll see here is the cycle of NADH to NAD+. That continuously gets recycled to continue the process. And last point here, it does occur also in muscle cells when we're working them out strenuously. Basically what's happening is the demand for energy is exceeding what can be produced aerobically. So temporarily we'll go into this lactate fermentation in the muscle cells. So glycolysis increases and pyruvate starts to accumulate because it can't be broken down fast enough in the Krebs cycle and the electron transport chain. So the muscle, it will remove the excess pyruvate, it'll convert it to lactate, which will get temporarily stored in the muscle cells. And usually you'll feel that as muscle cramps and muscle fatigue if enough of that lactic acid builds up. Then when oxygen is present again, usually through heavy breathing after exercise, the lactate will then get converted back to pyruvate, and then pyruvate will then be processed as usual through the aerobic pathways. The next type was the ethanol fermentation. Anytime you see it ending with OL, you know it's going to be an alcohol. So ethanol fermentation, this is an alcohol. So this happens in yeasts and some kinds of bacteria. They're going to convert the pyruvate to ethanol and carbon dioxide. All right, so here you have the pyruvate. It's going to form a 2-carbon compound, and then it's going to form ethanol. And we also see that carbon dioxide is also released as well. Okay. This is just a quick recap of anaerobic respiration. So anaerobic respiration, it does not completely break down glucose. It yields only 2 ATP molecules for every glucose molecule. So multicellular organisms, including us humans, we can only operate in oxygen-free conditions for just a limited amount of time because not enough energy is created in anaerobic respiration for us as multicellular organisms. Organisms that do rely on anaerobic respiration for an energy source are typically unicellular, so single-celled organisms like bacteria and yeasts. So recall that glycolysis, the starting point of respiration, can happen even in the absence of oxygen. So if there's no oxygen then present at the end of glycolysis, that's when the steps will then go into either lactate fermentation or ethanol fermentation. All right, quick review here of lactate fermentation. So it's occurring in some yeasts, some single-celled organisms, and some animal cells like our muscle cells when there's an absence of oxygen. So like I said before, when the products of glycolysis have exceeded the oxygen supply, pyruvate molecules start building up faster than they can be used. So during like a strenuous workout, the muscle cells will convert the pyruvate to lactic acid or lactate by oxidizing NADH to NAD+. That NAD+ will return again to be used in glycolysis again. And that buildup of lactate or lactic acid can cause muscle cramps and fatigue. Then when oxygen is available again, the lactate will get converted back into pyruvate, and then it'll go back into aerobic respiration inside the mitochondria. So ethanol fermentation, this was in some organisms like yeast and bacteria. They'll convert the pyruvate to ethanol and carbon dioxide by oxidizing NADH. Then that NAD plus will be returned again to be used again in glycolysis. All right, so in summary for anaerobic respiration, again, it's used in many industries like the food industry, the fuel industry, alcohol industry as well. So these single-celled organisms are valuable in the production of foods like bread, yogurt, fuels, and as well as alcoholic drinks. And the initial step always begins with glycolysis. Glycolysis can occur when there is no oxygen available. And then when oxygen continues to not be available to normally aerobic cells, then the process of lactate fermentation will occur. Glucose becomes oxidized and forms pyruvate and ATP. Pyruvate will then be reduced by NADH to form the lactic acid. Many cells, bacteria and yeast, they do not need oxygen to survive, so they can continue to function through the process of alcohol fermentation. So one good example is like if you left leftovers in a lunch container and it was sealed in a container for a few weeks, then you'll have the process of alcohol fermentation occurring. Glycolysis will break down glucose, which will provide the energy for the cells to do their work, and it'll produce ethanol and carbon dioxide. The container will start to bulge out because of this carbon dioxide gas being produced, and then you'll even be able to smell that smell of fermentation. Both lactate fermentation and alcohol fermentation will produce only 2 ATP molecules from the oxidation of glucose. So just like we saw when we looked at aerobic cellular respiration, it was much more efficient at producing ATP than aerobic respiration is. All right, let's take a look at some review questions here. Under what conditions does fermentation occur? It is the absence of oxygen. What are some common commercial uses for fermentation? So alcoholic drinks like beer and wine, vinegar, buttermilk, yogurt, cheese, breads, as well as replacing some fossil fuels to produce biogas and biofuels. What is the significance of the oxidation of NADH to NAD+ in ethanol and lactate fermentation? We need that to occur because that NAD+ will continue again to the process of glycolysis so that glycolysis can continue to occur in those cells. All right, so I'm going to pause here. Actually, we do have a video to watch, but I'm just going to finish the last 2 slides here. VO2 max and metabolic toxins. So athletes understand that process of cellular respiration to basically increase the body's ability to deliver and utilize oxygen so that the body can become more efficient in the production of ATP. So higher levels of physical activity can cause the accumulation of lactate or lactic acid because pyruvate is being produced by glycolysis and is not able to be oxidized by the Krebs cycle. So the point at which an athlete begins to accumulate lactic acid or lactate in the muscle tissues can be used as an indicator of fitness. And it can also be utilized as a tool for planning further training to improve performance. So there's a test called the VO2 max, which basically is the volume of oxygen used at maximal exertion. And it basically determines how well an athlete is able to use oxygen. So usually it's done in a sports medicine laboratory because it requires special equipment. And so, for example, in this picture, it's happening, they're doing it on a stationary bike, but it can be done on a treadmill. It can also be done on like a rowing machine. So during the test, the athlete will basically exercise to exhaustion while the heart rate and chemical composition of the air are analyzed, of the inhaled and exhaled air are analyzed, and then measuring the quantity of oxygen used by the athlete. Basically the athlete, they wear a mask to allow the inhaled and exhaled gases to be analyzed, and they're being analyzed for their concentration of oxygen and carbon dioxide. So then the values for the VO2 max are expressed as milliliters of oxygen used per kilogram of body mass per minute of activity. So milliliter per kilogram per minute. All right, so this technology, which is based on cellular respiration, helps athletes train and achieve higher levels of fitness. And then lastly are metabolic toxins. These are chemicals that can impair or disrupt metabolic pathways, such as photosynthesis and cellular respiration. So for example, a metabolic toxin can prevent the proper functioning of chloroplasts or mitochondria. So recall that chloroplasts, this is where photosynthesis is taking place, and in the mitochondria, that's where the cellular respiration is taking place. All right, so for example, pesticides and herbicides can interfere with the metabolic processes of organisms and then ultimately cause their death. But scientists have to use the scientific process to determine the harmful effects on humans, as well as the desired effects they want on the pests and the weeds. Continuing research on metabolic toxins can help scientists discover ways to inhibit cancer cell growth by creating toxins that can inhibit cellular respiration in cancer cells, specifically in the mitochondria, that would ultimately cause the cancer cells to die. So I'm just going to pause here and we'll watch the video on fermentation.